I request all of you now to please start. The lecture is starting. We are starting 10 minutes before, so that we can save time later. And today's lectures, there are two lectures today, as well as the tutorial as well. So it is on consolidated bioprocessing, and W will also highlight few of the achievements and research highlights what they have done. And I think in your handouts or in your study material, you may have got some papers also. So papers published by this group, you can read between the lunch time or like that, so that you will have some questions and discussions. So I welcome all you for starting the lecture.
Good morning, everyone. The topic of today's lecture is consolidated bioprocessing. Yesterday we learned how to use the bioethanol from biomass from cellulosic feedstocks according to the second generation of bioethanol. In the routine process for the second generation biorefinery, we have a number of operation units. The first is biomass preparation. I introduced it yesterday. And then we do the procurement of biomass with chemicals such as acid, such as sulfite chemicals for examination of lignin. And sometimes we use dilute alkaline. Alkaline is in general not as good as acidic agent because dilute alkaline is very difficult to recover to treat. Only the alkaline with high concentration that can be easily recovered in the biorefinery system. And the price of alkaline is relatively higher than the sulfuric acid. So I think it's better for us to choose acidic chemicals for the procurement. In addition, acidic chemicals can also digest a small amount of hemicellulose dissolved into small ankles, small fragments. So I introduced the acidic sulfide and acid treatment yesterday. Then the purchased solids were subjected to hydrolysis system. Hydrolysis system I showed you. It's a tank, it's a blending in which not only biomass but also the buffer and the inline. We use commercial enzymes or we use cellulase in our own lab. That's all okay. After that, we are going to fermentation. Okay. There are four different technologies. The first one is separation between hydrolysis and fermentation. It means after hydrolysis, we can do separation of solid and liquid. Only liquid is subjected to fermentation system because in the first generation biorefinery, it is commonly used. We are going to use immobilized saccharomyces, the yeast. Yes. Then after immobilization, it is better to use a clear hydrolysis liquor in order to keep the activity of the yeast. We call this process SHF. SHF means separate enzymatic hydrolysis and the fermentation. There is a separation operation between the the two units.
Switch on all the SC.
However, there's some shortcomings of the SHF process. First, it depends highly on the separation. And after medical hydrolysis, all the biomass solids they become into very short time particles. It is not easy for the separation of them from the liquid. And we have to add the separation facilities in the biorefining system. It's a lot of investment, and we usually consumes much more time because hydrolysis usually we spend 2 to 3 days, and the fermentation it also needs 2 days. So totally we need 5 to 6 days to produce ethanol from biomass. Then we have a good idea, use SSF. SSF means simultaneous saponification and fermentation. Previously, at the earliest stage, the yeast powder, the yeast is inoculated into the hydraulics system simultaneously with the commercial enzymes. They work together. They work together at the initial stage. But later, some experts found that it is not good because starting concentration of glucose in the SSF is very low, and the yeast cannot produce ethanol at a very high conversion rate. This image illustrates the SSF data from long times. Not only the conversion velocity, but also the productivity is very low than the typical SHF. And the total time consumed is also very long to reach a comparable level of ethanol treatment. So SHF has to be modified, has to be improved. There is a improved strategy. It is named as HHF. HHF means enzyme is loaded not at the initial stage, but at 12 hours later. 12 hours later means the hydrolysis. Okay, the enzyme has digested maybe 30% of the biomass, 30% of the cellulose, and the glucose concentration is higher than the initial stage. And the fermentation microbes can start the fermentation at a very relatively higher conversion rate. SSF is the HHF is better than the conventional SSF. Not only the ethanol teacher, but also the conversion rate is very good. But we still have a lot of problems. For SSF, we found that the enzyme and the yeast do not share the same optimal temperature. For the cellulose, optimal temperature is usually around 50 degrees centigrade. But for yeast, no, usually below 40 degrees centigrade. So it would be a problem. If we use a 40 degrees centigrade, the enzyme efficiency is limited. If we use a higher temperature, yeast does not work. So this is one shortcoming of SSF. Another problem is the ethanol inhibition. During the SSF, the accumulation of ethanol is very notable. It's very considerable. As the ethanol content arrives at reached 2%, the inhibition effect is very significant. So it is another problem. Then some researchers provided a new ideas, new concept is NSSF. NSSF means non-isothermal saponification and fermentation. It solves the first problem, the optimal the temperature difference between yeast and the cellulose enzymes. An SF design is like this. Separification and fermentation. They were performed into separate vessels, not together, separate. In the first vessel, in this vessel, biomass was loaded and enzyme was loaded, and the hydrolysis liquor they were transferred through into a new vessel. This vessel is a fermentation vessel. So the lignocellulosic column reactor, the working temperature is 50 degrees centigrade. And the influence in the column reactor, fermentation reactor, the temperature is 28 degrees centigrade. After the fermentation, the broth was pumped back into the hydrolysis column. The liquor recycles and in the circular process, and it was heated here, heated by a heat exchanger. And after hydrolysis, it flows back to the fermentation tank. There is a cooling. There's a cooling process. So the hydrolysis and the fermentation, they were performed in different reactors. It is named as NSSF. They were performed at different temperature. Another improvement is the SSCF. SSCFC means co-fermentation. Co-fermentation of hexose and the pentols. Because the subcommittee, we normally use the bio-type subcommittee does not ferment xylose and not ferment arabinose xylose, and so we have to do the engineering. Okay, I can engineer the subcommittee and let it has ability to co-ferment glucose and xylose. Another strategy is to use a co-culture. A co-culturing subcommittee with, for example, the xylose fermenting microbit, it will be very good. The ethanol production rate and the ethanol titer will be improved. Here's a table. The data is from John's work, and they use Cellulosic Ethanol, and this table is from the NSSF. Yes, they tried to do the SSCF and co-fermentation of glucose and xylose, achieved improved ethanol yield and achieved improved ethanol titer. Now let me introduce CBP concept. CBP concept means consolidated bioprocessing. This process, this technology combines the generation of digestive cellulose, xylulase, and other hydrolysis, and the sacrification of saccharides, and the fermentation of simple sugars into a single step. It means in one tank there will be at least two types of biocatalysts. One type is inline, digestive inline, and another type is fermentation. Okay. For a cellulose, for pure cellulose, there is no procurement, but for lignocellulosic biomass, there is a procurement. All of the materials can be deposited into the consolidated bioprocessing. And the microbit, we have three different types of microbit. The first type is natural microorganisms, including the bacteria and the fungi. Okay. The most famous bacteria for the CBP purpose is the Clostridium Clostridium difficile. For the fungi, we have white blood fungi. It is used for purification here. White blood fungi is Bacillium mixing. And we can also do the engineering of the supremacy, letting it display the cellulose enzymes on surface or secrete the cellulase into the fermentation culture. We can also use co-culturing. Co-culturing, we use another term is consortium. Consortium. Consortium usually we use cellulolytic microbiome and fermentation microbiome, and maybe a yeast, maybe a xylose fermenting microbiome. So finally, we got the ethanol. This process is very simple. Okay, it saves us a lot of investment on about the facilities. Okay, we do not need to do separation. Each step, we do not need to do separation. And the CBP process offers more advantages. First, it eliminates the use of commercial enzymes. We do not add enzymes, but in fact, in the practice, most researchers they still have to supplement the system with better glucosidase because microbes like Aspergillus niger, maybe the trichoderma, they sometimes they do not have sufficient activities of better glucosidase. But the total demand of commercial enzymes is significantly decreased. Another advantage is this process simplifies the biorefining of living cellulose. It saves the investment of facilities. This process also enhances the bioprocess efficiency. I couldn't believe that. I think it's slow. It's very slow because the enzyme, the microbes, it needs some time, many more times to produce the digestive enzyme. And the enzymatic hydrolysis rate is very slow at the first stage. And finally, it minimizes the total cost. Especially the white blood fungi.
Yes, you believe this one. Minimize the total cost?
Yes.
You believe?
I believe it. If we use
but it doesn't enhance the efficiency.
Efficiency is
not
good. I think so.
Even as per the principles of chemical engineering also.
Yes.
Biomass conversion and all that.
Yes. Not only the efficiency, but also the final yield of ethanol is more important. And we use white blood fungi. This microbes offers further advantages such as the purification. It serves the process of pretreatment. It means white blood fungi can produce the digestive enzymes for liquid degradation. So it will be very good. And some special species they co-ferment cellulose and xylem. We have this species, but the efficiency is not good because ethanol tolerance is really poor for this species. Here I make a list of the most commonly used CBP microbes. We have formicat, especially is Clostridium. Clostridium somomethylum ferments glucose cellulose, but does not ferment xylose and xylem. There is a very new research. They also prove that this strain can also utilize xylose, but requires several weeks or 1 month. Quite a long time. And it digests not only cellulose but also xylose. they produce ethanol from there. And only white portfolio, the Basidium like the Philobia, they can digest all the components in the biomass, including cellulose, xylem, and lignin. Next, let's come to the cellulolytic bacteria for the CBP purpose. Bacteria is very important because it grows very fast, and some species is very new, is newly developed, is newly isolated, and then discovered and characterized. So we have a lot of opportunities to do new research work from the bacteria. The first species is the Clostridium species. All of these the bacteria are gram-positive. They have bottle-shaped endospores, and they are anaerobic. Most of them are strict in anaerobic conditions. They produce alcohol, not only alcohol but also hydrogen and carbon dioxide and some organic acids. For example, this one, this is Clostridium cytobutylism. I studied for 5 years. It's a very good strain to produce alcoholic solvents from biomass. But its product is not pure. It has acetone butanol ethanol fermentation process. In this process, the products are very complex. This strain digests sugar, starch, and glucan, such as corn coba. So mostly I cultivate this strain. I use corn coba or corn juice. It degrades cellulose at a very slow velocity, very slow. It has cellulase including genes. Many genes they express cellulase, but the activity is not very high, very low. It has complete cellulose cluster genes, but not well expressed. No one knows why. A lot of new research on this topic. This strain has previously been used for producing acetone during the First World War, because at that time the oil biorefinery has not been established. It's the only best way to produce acetone from starch. The Weizmann developed this process. So this strain is also called as Weizmann organism. Recently, it is increasingly developed to produce butanol from dark starch and from biomass hydrolysis. However, the accumulation of butanol to content 1% to 2% will be very harmful to this strain. Very harmful. Butanol is a toxic chemical to microorganisms. It will destroy the picture of the cell and to maintain internal pH lower than cellular level, intracellular level of HE, and inhibits the glucose uptake. A lot of researchers they are trying to improve the accumulation level of the butanol during the cultivation of this strain. My lab will also do this work like this. Here I have a question. The Clostridium species are all anaerobic. So how can you cultivate this bacteria in the lab? Can some students answer this question? How do you do?
How you do the anaerobic culturing? Asking more. Any anaerobic culturing? How you?
This is the only question in this lecture.
Just anaerobic
Chamber. Around anaerobic chamber. Full of hydrogen, okay, full of carbon dioxide. Am I right? But if I do not have this equipment, what should we do?
Means if you don't have equipment, anaerobic component, how you will be growing an anaerobic bacteria? You are being taught general microbiology, how to grow an
I spent a lot of time, 2 years, to solve this problem. But it's very difficult.
If you need to get rid of oxygen, so how can you do that? It's very simple trick. They will answer right now. So it's a simple trick how to get rid of oxygen.
So how many of you have grown anaerobic bacteria in your life? Academic life, BSc, MSc. How many of you have grown?
They're all growing inside your muscles.
Okay.
So how is it growing there?
I will show you my method. I have 2 ways. The first method is use a glass roll or liquid bags. Okay, on the top, seal the top of the container. I separate the oxygen with a conduit.
Just by layering the surface by glycerol, he is able to generate those anaerobic energies.
See, this tube is, do you know what is its purpose? It's for counting the number of the of the bacteria. This method developed by my friend Mr. Lee. We work together. He has no background on microbiology. He has a background on business.
That's great. It means that anybody can try all these things.
Yeah. Well, we're very familiar with plate for counting the number. Plate is very good. Okay. But he told me that it is better to use the tube. To use the tube to do the counting of anaerobic bacteria. He used a dye, okay, to indicate the production of hydrogen. It's hydrogen. It's a very long story here. Here we have the colorful spot. One spot represents a column of strain of the bacteria. So we count the spots, maybe for example 14, and then we multiply the dilution factor, dilution times. So we get to know the number. It's better than the plate because if you use a plate, you have to put it into a special chamber. Not easy for a man have a background on business. And I developed another technology. This one, see, full of carbon dioxide. But why? I do not produce any carbon dioxide because I found that most of the anaerobic bacteria in the species of Clostridium, it produce organic acid. So I added the calcium carbonate into the substrate. Okay. As if there is generation of organic acid, the carbon dioxide will be released in this plastic bag. The plastic bag can tolerate pressure, high pressure of the gas. I use this method to produce important bacteria. This one, Clostridium fructifera. now I am one of the most important supplier of this bacteria. Use this method. I do not have a method. Because of method for an aerobic bacteria is extremely expensive.
So another simple way would be if you light a candle inside a closed chamber,
Yes.
all the oxygen will be used up by the flame,
Yeah.
and you can see that you can get rid of all the oxygen.
Yeah.
The conditions
It's also a very good method.
Yeah.
So sometimes if we use chemicals or other stuff or contaminate the substrate, we want to obtain very good cells from the cultivation.
So how long does it take to generate the anaerobic conditions, especially in this bag?
This bag is control. There's no
Catching
common combination, calcium combination. There's no combination. Yes.
So this is on its own. The swelling of the bag is once. So when the carbon dioxide is reduced, the bag automatically bulges up.
Yes. It usually requires only 4 hours to generate
This much
gas like this.
Yeah, exactly.
Yeah, everyone has his own method, I think so. Okay, next is a very famous strain. It's Clostridium solum. This strain is a thermophilic bacteria. It produces cellulosum. Cellulosum is a cellular machine. It's a very small machine. It assembles a lot of different hydrolysis on the surface using scaffold like this. And this bacteria has both abilities of degradation of cellulose and the production of ethanol. It highly depends on the efficiency of cellulosome. This is a microscopic picture of the digester of cellulose. This fiber, cellulose fiber, and bacteria like this, it absorbs on the fiber and produce some portions and walls on the surface. The cellulosome is multifunctional and multi-enzyme complex, assembled with a scaffold as in contents. Here is a schematic diagram. On the surface, there will be a CBM first. For each cellulosome, there might be a CBM. It is used to direct the orientation of the bacteria and absorb on the surface, on the cellular surface. Here I showed the structure of cellulosome produced by the Clostridium. The cellulosome is multifunctional, multi-enzyme complex. On each scaffolding, there are 9 repeat cohesion type 1. Type 1 connects the scaffolding with the enzyme. This is type 1. There are 9. This cohesion accommodates organizing cellulosic hydrolysis. And there's a type 2 cohesion docking. It is used to connect with the bacterial cell. For each cellulosome, there is 1 CBM. They share 1 CBM for the docking on the substrate surface. Here I list the genes for many Clostridium species. Many of them they have a lot of docking containing genes, cohesive containing genes, and they combine together to be a cellulosome. Especially this one, the ATCC strain, and
Cellulosome must be a photoenzyme consisting of many different enzymes which
Yes,
are
many
derived from
different
different genes.
yeah, from different genes, yes.
So this is a complex.
Yes, it complex, yes. But these two genes are for the docking and the cohesive. Yeah, I illustrate the metabolic pathway of the Clostridium. It has two branches. At the first branch, the pyruvate branch, there's an important byproduct of lactate. And at another branch here, there will be a production of carbon dioxide and the formate and the hydrogen. So directly fermented cellulose and other beta glucans to ethanol, usually we have a lot of different byproducts, especially the hydrogen. Okay. So that's why we use native Clostridium strain to digest this biomass to ethanol. The yield, the ethanol yield can hardly about 50% because the formation of different byproducts. So the research work on the Clostridium submotilum focuses on the metabolic engineering. Metabolic engineering, especially to decrease, to delete the genes for producing acid and hydrogen. In order to improve the ethanol yield, and some work are also focused on the improved ethanol tolerance. So we have two ways to do the metabolic engineering. The first one is carbon flux, to disrupting carbon flux. Another one is to disrupting the electron flux. Electron flux generally reduce the production of formate and hydrogen. And the carbon flux generally decrease delete the genes of lactate dehydrogenase, and it will reduce the byproduct lactate by 30%, and improve ethanol tolerance. But the second method is much more efficient. Recently, some experts also found that the Clostridium produces oxidase. This oxidase helps penetration of E9 of the cellulosome into the substrate. And there might be some other functions of this oxidase, but not clear yet. It is a very hot topic, research topic. And some of the oxidase they have active center composed of copper ions, very similar with lactase and some other copper ions oxidase. Another Clostridium species is Clostridium celluloborans. Celluloborans produces a cellulosome and also free cellulase, especially the A2-glucanase. With CBMs like fungal cellulases, they have very similar properties. The celluloborans and A2-glucanase also exhibited the xylanase activity, and it can serve as a good holocellulosic lytic candidate. So it can utilize not only xylem but also cellulose. However, this strain does not move. It's a non-mobile road line. This strain also has a limited capacity for production of bioalcohols, especially with the ADH and enzyme allows the butanol production and ethanol production together. This is a metabolic engineering effort. The third Clostridium strain is phytofermentants. Phytofermentants secrete individual enzymes. Rather, it does not produce free cellulose. The cellulosomes. It secretes individual free enzymes like example cellulase. It includes almost all of the cellulolytic hydrolysis for the biomass conversion. Not only the cellulase but also the hemicellulase. It goes on cellulose and the hemicellulase at the 37 degrees centigrade produces ethanol, acetate, carbon dioxide, and hydrogen as minor products. It is a very good candidate for the CBP purpose. However, it usually requires a very long time, for example, several days to accomplish the conversion of biomass to ethanol. And the ethanol titer is usually not higher than 10 grams per liter. Another type of bacteria is hemicellulolytic CBP bacteria. This type of bacteria, they are very good at the digestion of hemicellulose. For example, some anaerobic bacterium species previously known as Clostridium Cellulolytic. This species grows on the form of hemicellulose by secreting the xylase and ferment silane xylose glucose to ethanol, to lactate, to acetate, to carbon dioxide, and to hydrogen. So engineering of these strains will improve the final yield of ethanol by to 90%. We also have two other species. One is Cellulolitis and another is AK-17. I did not do study like this, so I have very little idea about these strains. Okay, here I showed some engineering efforts. Engineering efforts for AK-17. The first effort is the knockout of genes involved in the organic acid formation. It is very useful to reduce the formation of organic acids. Another effort is biofunctional alcohol and ADH genes. And someone also deletes this gene to eliminate the formation of hydrogen. Okay, this lecture comes to the end. Thank you. Do you have any question?
The thermoanaerobic is more successful in producing the non-process of CBP. You have used to bacteria, thermoanaerobic sediment prostration.
Which one is better?
Which one is the best?
Which one is better? medium or Clostridium?
Which bacteria, which species is better for maximum production?
Which one is better?
Better, yeah.
I believe
As per your experience.
yes, this one. I read a lot about this bacteria. First, it has a variety of hydrolysis genes. Almost for all of the components in biomass, including the happiness, including the arabinose, digestive yeah. Second, it can tolerate ethanol as high as 1%, and it produces acids and hydrogen as a minor product. The byproduct level is very much lower than Clostridium, the cellular. Yes, I think this one is better, much better. Yeah.
So this corn stover is you have purchased or you have used it for agro recipe as an agro recipe?
Yeah, it is. This work is not my work. It's from James' publication.
From basic.
From basic, yes. And the AFEX is a routine procedure method depends on the ammonia, treatment of ammonia. It's alkaline treatment. Very efficient because the crystalline shape of cellulose will be transformed during this treatment from cellulose 1 to cellulose 2. Cellulose 2 it has a much higher space between the microfibers, between the chains. So the enzyme is more ready to digest after the treatment. Yes, I tried the AFAX is very useful. It's very helpful. Yes.
One more query is there. This affects pre-treated column stover. This gene 2012, using with William University, they are working on lignocellulose biotechnology.
Yes.
Or they are working on pulp paper technology.
I'm not sure.
You don't know. They are not collaborating with your group.
No.
Okay.
You can also try this in the kidney.
We are planning that we should have the pre-treatment methods for our MC student. Because the pre-treatment is still many students they don't know. Direct pre-treatment nobody knows which will be suiting for what type of purpose. So we will try to have 2 projects this year. And we will be needing your help as well.
In last lecture, I introduced the dissolution of cellulose using organic liquid. Yes. Actually, we have another cellulose solvent. It's phosphoric acid. Phosphoric acid it works below 0 degree centigrade. It will breaks down all the inter and the intra hydrogen bonding in cellulose and produce very pure amorphous cellulose powder.
So you told us different configurations like SHF, SSF, SSCF, CBP. So in SSCF, you told us that we need to add enzyme and dose fermenting culture and exhaust fermenting culture. And similar is in case of CBP. Then what is the difference between SSCF and CBP configuration? SSCF and CBP. What's the difference between the two? Because in both these configurations, we are adding enzyme, we are adding pentose culture, we are adding exhaust culture.
Yes. SCF, in general, we use 2 microgrids. One is Cellulosic and another is PCB. If we use these two types of microorganism for the fermentation of glucose and xylose, it will work very efficiently. But both of them, none of them can digest the cellulose or xylene directly. They can only ferment sugars, simple sugars. So it is also called the half or semi-CBP. Yes, semi-CBP.
Because in CBP we are adding enzymes also for cellulose degradation and
Yes.
xylene degradation.
The level of cellulose is very important. The activity, the initial activity is very important. For SSF, it will be a very big problem if the 0 point time at the initial time there is no cellulose and there is no cellulose. Okay, the rate is very slow, very slow rate. You cannot detect the product from the growth.
Yes. And sir, what is HHF? What is the full form of HHF?
HHF is modified SSF. The time we colonate the yeast is not at the initial time.
Yes.
Maybe after 12 hours later. At that time, the sugar has a much higher level.
Sir, one last query. Suppose we get the CBP strain with us, apart from modifying it genetically,
Okay.
what strategies we can follow to enhance the yield of bioethanol production from
Um,
CBP strain? Without genetic modification, can we follow any other strategies?
Here I summarize the two methods. The first one is trying to disrupting the carbon flux. Another one is trying to disrupting the electron flux.
So this is something true metabolic engineering is suggesting what they have assigned. I think you are telling that metabolic engineering can sort out this type of thing.
Yes, can improve the quality of the capability of the stream.
And all CBP strains are anaerobic. They are
No,
not.
of course not. I will explain it in the next lecture. Because the oxygen requirement is different for cellulose production and different for the fermentation. Totally different. If we use
Yes.
if we use SSF strategy like this, SSF, okay, we use trichoderma resin to produce cellulose. It requires oxygen. But if we incolonate the yeast, the oxygen level should be very low. Okay, so they are different. How can you do that? Yeah, they're different.
I think the most important slide was the points where we can engineer the bacteria, the metabolic engineering, what we should change the carbon flux and the electron flux. So you should try to target those two areas because most of the research is now focused on metabolic engineering of these microorganisms because the native species of these microorganisms have their own limitations. So he very well summarized in that slide that which are the 2 points of engineering. One is to divert the carbon flux and the other is to divert the electron flux. So I think the participants should focus on that slide. You will also have a small exam over that. So I think definitely this slide is something which is currently the hot topic of research. Because most of the time people are working with either disrupting the carbon flux or the electron flux.
Even this metabolic flux and flux balance analysis, I told many times to all the students, they can apply in other research areas as well. If they target this thing, that will sort out most of the things. Right? Yeah, time.
Yeah.
5 minutes. Most of the students who are MSc students can note down in your notes because this is very important thing. And I hope with this type of research which he is doing, it is very simple also. You don't need not to have a lot of money to do that. Like he showed a very good experiment where anaerobic culture has been done. So, and that to a person from non-microbiology background. So yes, so it requires the skills you can do it in your lab also. So many times you have seen.
Otherwise, generating anaerobic condition like she's working on anaerobic organisms. Then she will be doing cell culture in that, and that whole system has costed about 5 to 6 lakh rupees. So these were two very good concepts of creating anaerobic condition. First, he discussed was by layering the media by cholesterol. Cholesterol is highly viscous liquid and it layers over the media and it creates deconnects the oxygen and it creates anaerobic conditions. So that's a very good idea. And the other one he discussed was mixing calcium carbonate along with the cultures, which because the cultures create some organic acids. And organic acids when it comes in contact with calcium carbonate it generates carbon dioxide. So the swelling of the bag was actually the generation of carbon dioxide. And that's how we could create those anaerobic conditions. So for the organisms, what he was discussing, Clostridium is a very important organism, and engineering that organism is the hot topic of research nowadays. How to engineer, and metabolic engineering is a term that you should all understand. And the pathway he showed in different slides, those pathways can be engineered to economize the production of energy. So, if the organism is losing its carbon by generating organic acids, so that means that's a waste. Right? We should divert that carbon in the generation of ethanol. So all the energies which are being utilized by the organism for different aspects to sustain itself, we can divert those energy, and the technology is gene knockout. And Dr. Kiki can elaborate more on gene knockout, how to do that, what is gene knockout.
We are left with the student, so we can discuss this gene knockout in next session, or we will we have to bind up. Okay, in the course of this session, yes, we can discuss the gene knockout. Yeah, we can discuss.
So did you use any tool, any computational tool to study the flux balances, the carbon flux and electron flux balances?
You guys know the questions.
Did you use any computational tool to study this carbon flux and electron flux balances?
Actually, no. Because I do not have a good background on the metabolic engineering. I am learning, studying. I learned a lot from Dr. Pumit's son. He taught me how to read the metabolic map and how to predict the product, predict the products from the defined content of the substrate. For example, from glucose to ethanol, glucose to hydrogen, glucose to lactic acid. I know how to do that. For gene knockout, I don't know how to do that.
Sir, I would like to add to it, if you will allow me. For this tool analysis, you can go for metabolic flux analysis, flux balance analysis with the MATLAB software also. It's very simple. You can do that also. You can measure all the carbon flux, all the electron flux. You only need to know what are the extracellular metabolites and what are the intracellular metabolites. You just add all the equations in the software, and software will give you where this flux is.
But we do spend a lot of time and a lot of cost to do the expression of genes in the use of picking in Cellulosic For example, we express the cellulase, fungal cellulase from tuna on the yeast surface or secreting in the secreting form. We are doing research like this in this way. I will share my research results to you in the next lecture.
Yeah, no problem.
It's another topic different from this lecture.
So thank you. So we can take another queries in the second session. So with this, we come to the end of the first session. Now we'll be having a break for 30 minutes, and we'll be reassembling here after 30 minutes. So the second lecture will be consolidated by processing 2. So any queries we can have in next session.